using the same technique, similar research directed at other surgical problems is possible. For example, we can elucidate how the presence of cleft palate affects the risk of facial bone fractures. First, let me explain the basic idea of the study. Suppose we have a very high tower. When strong forces work on the tower, it might bend to some extent. But let's assume a part of the tower is lost for some reason, and the tower becomes asymmetrical. We can surmise that when the same force works on the tower, it will probably bend more than when it was intact, and could even break. Thus, asymmetrical structures have greater susceptibility to external forces than symmetrical structures. Let's review cleft palate from this standpoint. For the presence of cleft palate makes the skull asymmetrical. As I explained with the analogy of the towers, asymmetrical structures are susceptible to external forces. Therefore, we can infer that cleft palate patients are more likely to develop facial bone fractures in accidents. I conducted a study to verify this hypothesis. First, as a preliminary experiment, I prepared dry human skulls and struck the orbits of the skulls by dropping a weight, just as we did in the verification experiment for blowout fracture. I used some of the skulls as they were. However, with others, I removed part of the maxilla to replicate the condition of cleft palate. Then I struck the skulls and observed what would happen. I found that in the skulls with clefts, fracture involved greater areas of the orbital floor than in intact skulls. This finding suggested that orbital floor fracture tend to be serious in cleft palate patients, supporting part of my hypothesis. However, experiments on actual skulls can be biased by subtle differences in the positions of the skulls. Furthermore, the experiment, which I did 9 years ago, would probably not be allowed nowadays for ethical reasons. So, some methods other than using up more skulls was necessary to provide further support for this finding. To perform theoretical proof, I produced three-dimensional dynamic models of normal skulls and cleft skulls, then applied forces to the orbital limbs of these models to conduct a dynamic simulation. The areas marked with red color are the areas where fracture is theoretically expected to occur. These red areas were greater for cleft skull models than intact skull models. This result confirms the findings of the experiment with actual skulls. Specifically, the difference in fracture patterns between intact skulls and cleft skulls is attributed to the stability of the minor segment of the maxilla. The minor segment is unrestricted in cleft skulls, so it bends greatly when struck, aggravating fracture. We have thus demonstrated that orbital fracture is likely to be more serious in cleft palate patients than in normal persons. We published this study in 2010 in Scandinavian Journal of Plastic Surgery. I am still interested in how the presence of cleft palate influences the patterns of facial bone fractures in the trauma. So I have continued studies with other types of facial bone fractures using much more sophisticated models than 8 years ago. accuracy has increased, and this is what the latest model looks like. Currently, I'm working on zygoma fractions. I strike the chip in the models and observe the results. I have found that as with the orbital floor, fracture patterns present big differences between intact skulls and cleft-palate skulls. This study is ongoing. I will analyze the results and will report it in the near future. Besides elucidating differences, structural analysis is also applicable to determine treatment strategies for facial bone fractures. So let's discuss zygoma fracture as an instance. In the treatment of zygoma fractures, the deviated bone must be repositioned to its original position and then fixed there. The fixation is usually done at one or more of the three sites shown in these pictures. To be more specific, these three sites are: first, the inferior orbital rim; second, the frontal process; third, the zygomatic-maxillary buttress. In performing fixation of fractured zygoma, securing the stability of the fixation is the essential issue, because if fixation is unstable, the zygoma will gradually deviate and develop secondary deformity over time. The simplest way to increase the stability of the fixation is to fix the bone at multiple sites. For instance, we perform fixation at all the three sites, we would be able to secure sufficient stability. However, if you want to fix at all three sites, you must make 3 skin incisions. The issue is the more fixation we conduct, the more scars are left on the patient's face. So the fixation sites should be minimized. Aesthetically, it could provide sufficient stability with fixation at only one site. That would be ideal. Then, which of the 3 fixation sites can provide the best stability? We need to prioritize. To clarify this question, we produced 3-dimensional skull models and separated the zygoma to replicate zygoma fractures. Then, we reconnected the separated zygoma with mini plates and screws at the frontal process, inferior orbital rim, and zygomatic-maxillary buttress, respectively. Then, downward force was applied to the arch of the fixed zygoma, simulating mastication. By using structural analysis, we calculated the stress occurring around fixation screws. Greater stress around the screws indicates that bone absorption is likely to occur there, and the screws are likely to loosen. So the greater the periscrew stress is, the more unstable the fixation is. It was found that stress was greater in the order of inferior orbital rim fixation, zygomatic-maxillary buttress fixation, and frontal process fixation. The instability of fixation decreases in this order. In other words, the zygoma is most stable when it is fixed at the frontal process and least stable when it is fixed at the inferior orbital rim. Therefore, when we need to select one of the 3 sites for fixation, we should select the frontal process as the fixation site. The second best is fixation at the zygomatic-maxillary buttress. Sole fixation at the inferior orbital rim should be avoided. Our technique of structural analysis is very useful to clarify questions in many fields of plastic surgery. Let's move on to a completely different field, keywords. The word keloid originates from a Greek word meaning crab. It is called so because of a similarity to the shape of the crab. In particular, keloids on the chest tend to present the typical crab-like shape. Although the morphological similarity of keloids to the crab is very well known, nobody has ever succeeded in explaining why it takes the crab shape. Perhaps structural analysis can come to the rescue. Here, we call the relationship between stress and hypertrophic change of scars. When the body moves, fibroblasts inside or around the scar are mechanically stimulated, activated, and produce collagen. The collagen accumulates and presents hypertrophy. Therefore, by evaluating stress distribution patterns, we should be able to predict how a scar will grow. When high tension works on the scar, it gradually presents hypertrophy. Based on this principle, we performed an experiment. First, we obtained surface data of human chest. This can be done using three-dimensional scanning of the chest. After obtaining the surface data of the chest, we processed the chest into three-dimensional models. Then, by replacing a part of the chest region in each model with hard tissue, we produced models carrying chest scars. As the chest moves during respiration, the skin is stretched and the stress occurs around the scars. We calculated the stresses and evaluated their distribution patterns. The red area indicates the zone where intensified stress occurs. Observation of stress distribution patterns reveals that stress concentrates at the ends of the scars, meaning that fibroblasts are very likely to be stimulated there and to cause local hypertrophy. Given the relationship between stress and scar growth, we can surmise that where stress concentrates on the scar, it will develop hypertrophy. Therefore, if stress concentrates at the ends of the scar, fibroblasts are stimulated there, and the scar develops hypertrophy there. So initially straight and horizontal scars develop hypertrophy at their ends due to stress concentration, gradually transforming into shapes similar to bones. For the new condition after the plasmid formation on bone-like shapes, we repeated the same simulation and calculated the stress around the bone-like scar. We found that much higher stresses work on the protruding ends of the scars, the part indicated with the triangular arrows. The scar develops hypertrophy there again and further grows at these parts. Eventually, the scar grows into the X-like shape shown in the right below figure. Thus, we can explain why keloids tend to present shapes like crabs. In 2014, we reported this hypothesis in the Journal of Medical Hypothesis.